First, I would like to introduce myself. I'm a clinical pharmacist at Children's Cancer Hospital, Egypt 57357. And my journey with pediatric cancer started in 2008 when I joined the hospital. And thanks to the Fulbright that gave me an opportunity to move one further step and to study at the US at the University of Tennessee and have my thesis work at St. Jude Children's Research Hospital, which is a pediatric cancer hospital based on donation, same as 57357. So I will try to go as fast as I can, but it's a little bit long, so that's the thesis title. It's entitled Somatic Mutation Detection in Leukemia Derived Circulating DNA, and the aim was to test its utility in monitoring clonal dynamics and disease response in pediatric patients with acute lymphoblastic leukemia. In brief, pediatric acute lymphoblastic leukemia comprises 20% of the cancers in patients aged below 20 years. And as we can see here, the survival improved from less than 10% in the 1970s and increased up to 95% in the 2000s. And this was mainly due to using combination chemotherapy and tailoring the therapy based on the response to the chemotherapy and based on the biological and clinical features of the disease. So here is a little bit about the biology of leukemia. First of all, A shows a leukemic cell with one genetic alteration or an initiating event, as we call it. This then is followed by a number of somatic mutations and genetic alterations, and it keeps going on, so only one of the clones keeps expanding, so we have one clone present in a higher frequency than the others. This is usually the dominant clone, the one that we can detect at diagnosis, but the real fact is that at diagnosis we usually have all these number of cells that are genetically different, and we cannot detect them all by the current methods. So why is that important? It's important because many of the patients relapse despite this improvement in treatment. So when we come to a relapsing patient and we see the genetic makeup of the cells, we don't find that it's the same predominant clone that was detected at diagnosis. However, it's one of these subclones that was present initially at a low frequency. So that's how these subclones increase dramatically throughout the therapy, and then it comes to a high frequency that leads to the relapse. Why is it hard to follow this throughout therapy? Mainly for two reasons. Because the main diagnostic test for detecting leukemia is a bone marrow biopsy, and as you can see in the picture, it's an invasive procedure that requires usually general anesthesia for children. So it's hard to keep doing it over and over again. And another drawback of this is that we can only detect the cells that come out of the needle, so we cannot detect all the population of the cell that's present. Another drawback is that with the current techniques for detection of the cells, those techniques are mainly present on the surface markers, the immunophenotypic markers, or the genetic markers of the cells of the predominant clone, so other cells still go undetected throughout therapy. So I will here give a very brief idea about the circulating tumor DNA and why it's presenting a new hope for those patients. So cell-free DNA and circulating tumor DNA are short fragments of DNA that are released into the human blood when the cells die, either by apoptosis or necrosis. So, and these cells are coming from all the cells in the body, either they are normal cells or tumor cells, and from different types of tumor cells as well. So what's really interesting about those short fragments is that they can display the whole range of mutations and genetic alteration that's presenting in different cells of the tumor. So as we can see here, this gets us over the drawback of bone marrow biopsy. And when we have a blood sample, we can take DNA from different types of cells, so we can assess the heterogeneity of the disease. This has been implicated in a number of cancers before, as lung cancer, where we can see the change of the mutation frequency and the change of the clones throughout therapy when we change from one drug to another. and it has been implicated also in some hematological diseases like myelodysplastic syndrome, where we can follow the burden of the disease and be able to detect early relapse. As for my thesis, what we were doing, we aimed to see if this can be applied to pediatric patients with leukemia or not, especially that this DNA is present in a very low amount in nanograms, so it makes it harder compared to solid tumors. So we utilized a next-generation sequencing technique to test two things, to test whether we can detect different clones at the diagnosis of the leukemia and whether we can follow these clones throughout therapy. We use the next-generation sequencing approach, and this allows us to detect different cells at different time points, as shown in different colors here, and compare the frequency of each of them to the whole reads of the gene. So we had this on new patients for the prospective studies, on patients following the new protocol at St. Jude Hospital, which is a total 17 protocol. We were studying the patient in the first 42 weeks of therapy, 42 days, which is 6 weeks of therapy. This is called induction therapy. And this phase of therapy usually aims at getting patients into remission, which means that they don't have an active disease by the end of this phase. So we were able to have 6 time points and to analyze 6 time points for each patient. We isolated the cell-free DNA at first, and it's interesting to notice that we only used 1 milliliter of plasma, which makes it easy to use. And then we had adapter-ligated libraries, and then we were able to target or to capture those genes that were involved in leukemia and were known to cause relapse or therapy resistance. And then we used to read this with the next-generation sequencing. This was how our lab generated the list of genes. We went into a database and saw the frequency of each gene and whatever mutation that occurred in more than 3 patients, and had a link to any of those pathways, like an oncogenic pathway, tumor suppression pathway, drug metabolism genes. That was a CRISPR-Cas thing, or another gene panels we incorporated into the research. So what about the result? We were successfully able to detect all the mutations that were present at diagnosis, and this was standardized against the bone marrow samples. That's already done as a routine for the patients. So for all those clinically relevant mutations, we were able to detect them in the circulating tumor DNA. And also we were able to follow at these time points how these clones rise and fall throughout the therapy. Those were different patients. So coming to the conclusion, we finally had something that would be a non-invasive biomarker to follow these patients throughout therapy, to detect their heterogeneity at diagnosis, and to follow them thereafter. Why is that all important? It's important because whenever we detect those clones as early as we can and they have therapy-resistant genes, we can modify therapy accordingly and prevent them to reach frank relapse. Thank you.